Good evening, all.
Good evening, sir.
Good evening,
Am I
Mr.
audible to you?
Yes, sir.
Right. So today class is protein biosynthesis. Already I explained briefly in last class what is genetic code, what is protein synthesis, what are involved in protein synthesis. Right. Is it visible to you?
Yes, sir.
Right.
Yes, sir.
So what is protein synthesis?
It's a transcription of protein, sir. How protein is made. It's how protein is made.
What is central dogma? Central dogma. Central dogma, nothing but synthesis of protein. It is carried out by two mechanisms. One is transcription, another one is translation, right? In transcription, what happened? From the DNA strand, RNA, mRNA prepared. In translation, from the RNA strand, protein strand prepared. So before that, we learnt about genetic code, triplet codons, how many codons are there, how they are formed. So how many amino acids codes for each codons. There are total 64 codons. These are codes only 20 different type of amino acids, right? Hello?
Yes, sir.
Right. Central dogma means synthesis of protein by transcription. The first step is transcription process, DNA to RNA preparation. This is highlighted place. This one transcription, and the second step from the RNA to protein formation, translation. Right? Let's discuss one by one. First one, transcription. Right? Is visible to you?
Yes, sir. Yes, sir.
Right. Transcription is the process by which the information in strand of DNA is copied into new molecule of mRNA. So DNA information copied to mRNA. This process called as transcript. Here the transcript DNA information transcript to the mRNA. This process called as transcription. Right? In transcription process, the main requirement from this mechanism, one is RNA polymerase and magnesium or manganese cations. These are the main requirements for the RNA transcription. There are two types of transcription: prokaryotic transcription and eukaryotic transcription. They are more or less same, but there are difference in the first initiation mechanism. Rest of the elongation and termination almost same. So anyway, in transcription mechanism, 3 stages are found. They are first one, initiation stage. Second one, write down this. Please write down.
Yes, sir. Taking it down, sir.
Right. Initiation, second elongation, and third termination. These 3 stages in prokaryotic transcription and eukaryotic transcription. Right. So first of all, definition clear about the transcription. The simple transcript of DNA information into the mRNA. That is this process called as transcription, right? Let's see the transcription process in general. Is it visible to you?
Yes, sir.
Okay. For example, here, let me take whiteboard. This is double-stranded DNA, right? Adenine, thymine, guanine,
One.
cytosine, guanine, thymine, adenine, whatever it may be. So these are complementary strands, each other complementary strand. Here triple bond, guanine, cytosine always triple bond, and thymine,
Double.
adenine double bond. Right. This is 5 and 2, 3 and anticod. This strand, sorry, 3
3,
and
5.
5. Right.
Yes, sir.
For example, this red strand is mRNA. Now mRNA complementary to here adenine, right? So what is opposite strand?
A it will be,
DNA,
sir.
sir. DNA strand.
A it will be, no, sir?
Adenine codes in
Adenine.
RNA.
Thymine
in
always
DNA.
space with uracil.
Right. Enough. Uracil, guanine, cytosine, carbon.
Cytosine. Sir, C pays with G, right, sir?
What?
Second.
Right. Okay, right. Okay.
G, sir.
G.
G,
G pays
C.
with C.
G, C,
Adenine
A,
pays with
U. Uracil.
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 Right.
Yes, sir.
Oh, 1 minute. Here the first one actually, thymine. Is it thymine, right? Hello?
Yes, sir.
Thymine codes adenine common. Adenine common. But instead of thymine, uracil. Okay?
Yes, sir.
Right. So this information transcript to the mRNA strand in the form of AGCU. So this AGCU complementary to this strand nitrogen bases, right? Okay. Hello?
Yes, sir.
Right. Now this strand called as template strand. Template strand or antisense strand. Template or antisense strand. This one sense strand. It's clear?
Yes, sir.
Right. So this is, first of all, this is 5N to 3N. This is completely double stranded DNA, right?
Yes, sir.
Double stranded DNA. In double stranded DNA, anyone act as a synthesis for mRNA strand, right?
Okay, sir.
So which is complementary strand called as template. Complementary to the prepared mRNA, that is called template, right?
Okay, sir.
Now, was that one clear?
Yes, sir.
Right. Now let's see the initiation. Already discussed about the initiation requirement RNA polymerase. I will explain later RNA polymerase mechanism. And second, this for activity of RNA polymerase, here they require magnesium and manganese cations. First, initiation. Just wait. Okay. First step, initiation. The transcription is initiated by RNA polymerase as a holoenzyme from specific point called promoter sequence. The promoter sequence very important for initiation of transcription. It is initiated when holoenzyme attached to the exactly promoter site. Here the promoter site, some sequences are there. Some consequence. Hello? Hello?
Yes, sir.
Just wait. I am in secondary class. Just wait 1 minute. హలో
Yes,
sir.
sir.
ఎస్ సార్
రైట్ మీకు అన్ని క్లాసెస్ ప్రాపర్ గా అవుతున్నాయి కదా C1 C2 C3
Yes, sir.
Yes, sir.
ఎనీ ప్రాబ్లం
No, sir.
రైట్ ఓకే రైట్ వేర్ వి ఆర్ ఇనిషియేషన్ సో ఫస్ట్ ఆఫ్ ఆల్ వాట్ ఈస్ హోలో ఎంజైమ్ what is promoter sequence, right?
Holoenzyme is nothing but which has protein part and non-protein part, sir.
Okay,
Promoter site is the region where RNA polymerase goes and binds on it first time.
right. Promoter sequence,
Promoter sequence is the region where RNA polymerase goes and binds to for initiation.
for initiation, okay, but
Hmm.
here the special sequence, here the special, can I go for whiteboard?
Yes, sir.
Hello,
Yes, sir. Yes, sir.
okay, right. Say
నిర్మిశ్రుగా ఇప్పుడు
Clear? Hello?
Yes, sir.
Right.
Yes.
So be careful. First what in initiation, transcription is initiated by RNA polymerase, right? RNA polymerase, holoenzyme, coenzyme, two different enzymes are there. I will explain what is coenzyme later. So this is only the matter of subunits. Protein, as you said earlier, protein and non-protein part subunits. All together, holoenzyme. If one it is missed, it is called core enzyme. First, let's see the holoenzyme. RNA polymerase holoenzyme. This one. Here, actually we can observe here only sigma factor, right? Here, apart from the sigma factor, other factor alpha, 2 alpha, beta, beta 1, omega. These are all including with this sigma factor.
Sir, these are all subunits of RNA polymerase, sir.
Yes, RNA polymerase subunits.
Yes,
If you
sir.
take RNA, actually no whiteboard ఎందుకు problem అవుతుందండి. Just wait. Put in the problem. Is it clear?
Yes, sir.
Yes, sir.
Actually, this is RNA polymerase enzyme, holoenzyme. RNA polymerase. This thread-like structure, this is the DNA strand, okay? Right. This RNA polymerase made up of sigma
Five
subunit, beta 1, beta, are you follow this cursor?
Yes, sir.
Right. And alpha, 2 alpha and omega. Now this is exactly bind with the promoter site. This region where it is bind, that region called as promoter site. Okay.
Yes, sir.
This region promoter site called as promoter. Promoter site, right? Okay. In promoter site, two sequences are there. Minus 35, minus 10. Minus 10 region in promoter site. Minus 35 and minus 10. This is the starting point. Plus starting point. Let me change color. This is the blue color mark. Are you follow? Hello?
Yes, sir.
Yes, sir. Yes, sir. Yes, sir. We are
Right.
following.
This is start point. Start point means start point for what?
For initiation.
Initiation before starting. That means you're starting. What is started?
Synthesis of protein,
Right. No, not a protein. Synthesis of mRNA.
mRNA. Okay.
Okay. Now synthesis of mRNA start this site only. But recognition site, promoter site, just far away from the starting site. Starting site plus from right side. This side +1, +2, +3, +4, +50, 60 like that. Okay. Left side. This side minus. This side minus. This is very, very important in transcription. If you're not able to understand about the start point, left direction, right direction, minus 35, minus 10, you can't understand about transcription process. In transcription process, the most important stage is initiation. Be careful with this.
Okay, sir.
Okay. Right. First of all, remember initiation factors. Already told you initiation main factor RNA polymerase. This is RNA polymerase. To associated this magnesium, manganese, ions, all manganese cations also involved in this. Right. Later, now just before that, we should aware about the some recognition places and promoter places and starting site. And the most important one is downstream and upstream. You should know aware about the downstream and upstream. Left side, from the start point, from the start point, left side minus value, that is called upstream. Upstream. From the start side, right direction, plus value, that is called downstream. Clear this one? Hello?
Yes, sir.
Upstream, downstream also clear.
Yes,
Right?
sir.
Okay. Now, what are the consensus sequence? For recognition of RNA polymerase, there are number of sequences are there in DNA. But where exactly RNA polymerase bind? Where exactly RNA polymerase bind? To binding it, there are some special sequences are there, right? The special sequences are called consensus sequences. These are called as consensus sequences, such as these consensus sequences. For example, let me write here. Consensus sequences. The consensus sequences in minus 35 region. The special sequence here, TT GACA, thymine, guanine, adenine, CA. And minus 10 region, let me change color. T A T A T. So this is minus 10 recognition site. This is minus 35 recognition site. Right? In minus 35, these both are present in promoter region. But minus 35, actually the first recognition minus 35. Here the open, that means the DNA strand to the double helix DNA strand separated from the minus 30. Okay. First bind, RNA polymerase first bind at minus 35, later closed at minus 10. After that, after minus 10, next year the minus 9 or minus 8. Here this double helix strand unwind. Why it is unwind? Is it possible to prepare the RNA strand if it is in double helix form? In natural form of DNA, double helix
No,
like
sir.
this, like a thread.
No, sir, it is not
Is it
possible.
smooth? Is it smooth to preparation of mRNA?
No,
No,
sir.
That's why it should be unwind. The unwind after attachment of RNA polymerase at the promoter site. In this first recognitions, minus 35, mostly minus 35 region bind. Later bind at minus 10 region. After that, unwind of DNA helix. Right? And this minus 10, this sequence was discovered by Pribnow. That's why this box name called as Pribnow box. Pribnow box. Pribnow box, right? In the same that means the same region eukaryote discovered by Huggins. That's why in eukaryote Huggins box are called. Right? Is it clear first?
Yes, sir.
Right. In most prokaryotic promoters, the actual sequence is not a TT, TA, TAAT. Another concentric sequences also there, which is approximately at -35. Right? So two consensus sequences in promoter region, one at -10, another at -35. Okay. These two consensus sequences very important for initiation of transcription. Are you clear so far?
Yes, sir. So
Right.
this consensus region is the place where they are binding. Is that so?
Yes, yes.
Thank you, sir.
Exactly. And the role of the subunits. Let's see the role of subunit here. Some sigma 2 alpha, mainly the 2 alpha recognition of site, recognition of consensus sequences. Beta 1 binding DNA, and unwind DNA. Bind DNA as well, unwind. Already told you after initiation, attach at -10, later right. Beta 1 unwind, attach DNA and unwind. The most important factor sigma, attached promoter site. Sigma 1 or initiation for, this is the main factor for initiation of transcription, right? So mainly the sigma factor bind at promoter site. Sigma factor bind at promoter site. So these are the important of the RNA polymerase subunit. If it is eukaryote, in eukaryote not only one polymerase. In eukaryote there are 3 different polymerases involved in transcription process, such as transcription 1, RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 for rRNA, 2 for mRNA, 3 for tRNA. So this is different. But what happened in prokaryote, all type of RNA prepared by one type of RNA polymerase. Okay, that is the difference between eukaryote and prokaryote. Remember, in prokaryote, eukaryote 3 different RNA polymerases. But in prokaryote, only one RNA polymerase there. But initiation same, the same steps: initiation, elongation, and termination. Is it clear? Initiation clear?
Sir, here
Yes,
it
sir.
is Prigno box, and in eukaryotes it is Higgins box, right, sir?
Yes, that's it.
Consequences.
Yes. Okay. Right. And for initiation, 3 steps. Again, initiation 3 steps. First, closed complex. Second, open complex formation. Third, tertiary complex formation. Simple. The first one, closed complex. This is called closed complex. Closed complex means, just wait where it is. Right, okay. Closed complex means if the promoter site, this promoter site completely bind with holo enzyme of polymerase. This is called closed complex. Now open complex means what is your open? What is second step? First step clear. Already told you the open complex also. Open, what is open complex formation? What opens here? After attachment RNA polymerase, what opened?
The DNA
DNA
2
strand.
strands
Right.
open, sir.
DNA 2 strand unwind.
Unwind.
That is called open complex formation. Second one, tertiary complexion. In tertiary complexion, same after open complex, after moving 10 base pair, after movement of 10 base pair, this RNA polymerase moving towards, remember, RNA polymerase move towards downward or backward or upstream or downstream.
Downstream, sir.
Remember, always downstream. mRNA preparation also downstream, right? While it is moving toward the downstream, after 10 base pair, after formation 10 nucleotides of RNA, immediately this sigma factor of RNA polymerase removed out.
Yes,
Sigma
sir.
factor removed out. Now this is called as core enzyme.
Yes, sir.
Core enzyme. Now this is called as a tertiary complex. Almost tertiary complex elongation start, right? When the tertiary complex formed after movement of the RNA polymerase towards the downstream, right? After 10 means exactly reach the start site. When exactly reach the start site, then only here the sigma factor removed out. Now elongation started. After tertiary complex formation, elongation of transcription started. Right? Is it clear? Hello? Hello?
Yes, sir.
Is it clear? Any doubt?
So trying to understand that, sir. Could you repeat that once again, sir?
Which one?
This before the elongation, sir.
Before elongation, you said me, what are the stages? Again, I will explain. I have explained 3 stages.
Yes, sir.
What are those? First one?
Initiation.
It is binding, sir.
I am
Closed
asking in first stage of initiation. Again, 3
First is
sub-stages are there.
binding.
Sir, closed promoter complex, open promoter complex, tertiary promoter complex.
Right. Closed one, open one, tertiary one.
Tertiary. Okay, sir.
Closed one. What is closed? What is closed here? First,
The DNA
Sorry.
strand, sir.
The
DNA stand, not only DNA stand exactly promoter site closed by RNA polymerase, right?
Yes, sir.
So that is closed complex. Closed means attached at promoter site. That is the
Yes,
first
sir.
point. The blue color part, this is RNA polymerase, right? With subunits, okay?
Yes, sir.
This is promoter site, already draw this one. Promoter site. Now this
Yes, sir.
promoter site completely closed. Completely
Yes.
occupied with a RNA polymerase. Now this one called a closed complex, okay?
Yes, sir.
What is second stage?
Open.
Open. What opened?
The DNA will be unwind.
Right.
The DNA will
Unwinding of DNA is called open complex formation.
Okay, sir.
Next, what is third one?
Dissociation of sigma factor after 10
First name.
nucleotides.
After
Tertiary
mechanism.
promoter.
Right, tertiary
promoter
complex formation. Now tell me the mechanism.
Sir, sigma factor get dissociated from the
When sigma factor dissociate,
from the RNA polymerase enzyme.
when it is dissociated from the RNA polymerase,
After 10 nucleotides formed.
right?
Sir.
After 10 nitrogen bases moving toward the downstream, immediately the sigma factor released out. Now this is called tertiary complex. Now it is followed. What is the next stage?
Elongation.
Elongation.
Right?
So that means
That's
the tertiary complex is the formation of 10 nucleotide nucleotides.
Yes.
Huh?
Yes.
So that is the important one.
Yes.
Okay.
So, sir, in initiation, only 10 bases only formed, right, sir?
Right.
Okay, sir.
Next one. Right. Now second stage, elongation. Are you able to see? Hello?
Yes, sir.
Yes, sir.
Right. Right. I think initiation clear.
Yes, sir.
Right. So in elongation, after synthesis of RNA, more than 10 base pair. As I said earlier, more than 10 base pair. Up to 10 base pair, that means when reach the 10th base pair, immediately the sigma factor released. The sigma factor is ejected and the enzyme move along with the 5 and 2, 3 and direction, continuously synthesizing RNA. Always the new strand synthesized here, not only mRNA, in DNA synthesis also, newly prepared strand always 5 and 2, 3 and remember it, always 5 and 2, 3 and. Okay. So this elongation continues towards 5 and 2, 3 and at which down or upstream,
Downstream.
always in downstream, right? An elongation of RNA is catalyzed by the RNA polymerase core enzyme or holo enzyme.
Core enzyme, sir. Sigma factor
Right.
already dissociated.
Right. We can compare here this pink colored sigma factor. After formation tertiary complex, sigma factor released. Now here no pink color. That means pink color means no sigma factor, right?
Yes, sir.
Hello? Clear?
Yes, sir.
Now only the core enzyme move towards 5 and 2, 3 and direction. This direction. This direction. Always here given 5 and
3.
and here 3 and direction. So this process called as elongation. Right? After initiation, RNA polymerase moves downstream along the template. Along with template, moves towards the downstream. In bacteria cell, 37 degrees centigrade, about 40 nucleotides are added per second. In elongation process, per second, 40 nucleotides, only 40 nucleotide added to the RNA, right? And whereas in DNA synthesis, more than 1500 nucleotide per second in eukaryotes. In prokaryote, only 40. What about in eukaryote? In the same in elongation, 1500 and nucleotides, 1500 nucleotides added per second. As the transcription moved down, the DNA template, it generate positive supercoiling head of the transcription bubble and negative supercoiling behind it. What is positive supercoiling and negative supercoiled. What is positive supercoiling? Positive supercoiling means what is supercoiling? The DNA, it is, this is DNA strand. Always this is supercoiled like this. This is positive. Okay, right? Unwind negative. Unwind is negative. Here the 5 and 3 and direction DNA prepared, right? But behind it again rewind. Are you follow my cursor? Hello?
Yes, sir. Following the cursor, but not able to understand what it is, sir.
Do you know the DNA natural structure?
Yes, sir. It's a double helix. It's twisted.
Double helix. Double
Yes.
helix. This is the double helix section, right?
Correct.
It is formed by supercoiling nature.
Yes, sir. It's
Supercoiling
twisted.
nature. If the supercoiling nature positive, these are again rewind. Negative, unwind. Remember
Okay.
these two things only.
When it's unwinding, it is negative.
That's it.
Okay. So when it is winding itself, it's a positive.
That's it.
All right, sir.
Clear?
Yes, sir.
Right. So anyway, it goes along with towards 5 and 2, 3 and direction up to reach termination. Up to reach termination. Now the third stage termination. Is it clear? First one?
Yes,
Elongation
sir.
clear?
Yes, sir.
Right. Now termination. In termination mechanism, transcription is terminated due to specific sequence in terminator DNA. This specific sequence terminated by row dependent or row independent mechanism. Let's see what is row dependent, what is row independent. So anyway, what is termination in simple language? Terminate the DNA process, sorry, RNA synthesis process. During elongation, always the new nucleotide added towards the RNA, and it is goes on. But when special sequences are formed with two mechanisms, row independent, row dependent, termination takes place. Let's see what is row and without row. Okay. Right. In row independent. So this is row dependent. Are you able to see?
Yes, sir.
Right. So this is along red color mRNA strand elongation towards 5 and to 3 end. But here this pink color, this is called row factor, row protein factor. When the row protein factor bind to the DNA, immediately the elongation process stopped. Elongation process will be stopped and the prepared mRNA released out. Is it clear?
Yes, sir.
For example, this is the site. This is the site. This is the site for termination. Now here, row factor bind. Now it is block the row factor. Look, actually, this is blockage area. It's block the RNA polymerase. When the row factor, it is look like a just bend like structure like this. Actually, this clear structure like this. So now here this is RNA polymerase move towards downstream during elongation process. Now it is reached towards the profactor. Can it move forward? Can it
No,
move forward?
no.
Right. Then this prepared RNA released out. Now transcription complete. So this is DNA dependent or independent? Sorry, row dependent or independent?
Dependent, sir.
Row dependent factor. Right. Let's see second one.
Sir, row factor, what is row factor, sir? Is it from RNAs or DNAs itself?
No, actually, row factor in present in bacteria cell.
Okay, sir.
Bacteria cell, it is exactly where the rich A and uracil, repeated
Mhm.
A uracil sequences are present. There the row factor bind.
Okay, sir.
Then the automatically elongation stop.
So that means the row factor is only for stopping the process, sir?
Yes.
Elongation.
Elongation. To
not
terminate the
initiation.
elongation.
Yes.
Okay.
And without row, sometime what happen here? Just wait.
The structure looks like the zip, sir.
Which one?
The transcription, sir. It looks
Okay.
like the zip, the chain,
The
you know. It's
mechanism,
like,
the entire,
yeah.
entire mechanism. Okay. This is independent. Are you clear?
It's looking clear, sir.
Right. Now this is actually this mRNA strand. 5 to 3 direction. mRNA strand. The when formation, the repeated uracil like this, poly U site, and followed by GC rich, What happened in GC rich? Guanine adenine triple bond. Due to the triple bond nature, these are firmly attached each other. As a result, the loop formation within the RNA, within the mRNA, where the GC rich area present, automatically due to the attraction of guanine cytosine, immediately the loop formation formed. Actually, this is first actual before formation loop, hairpin like loop. This is mRNA strand, right? Uracil, and guanine, cytosine somewhere, cytosine. Here again uracil. But this bond formation, firmly the bond formation between guanine and cytosine. That's why immediately this straight line mRNA slightly form like hairpin like. Where the hairpin like formed, automatically guanine and cytosine formed. Guanine, cytosine. guanine, cytosine. Here uracil, right? Hello,
Yes, sir.
correct. So here only guanine, cytosine attached each other. As a result, this loop-like hairpin loop-like structure formed within the RNA structure. Now, immediately what happened here? This RNA polymerase cannot move forward due to the hairpin formation of DNA structure. Already folding formed. Is there any row involved?
No.
Same row look like a hairpin like. But without row form involved, automatically within the RNA, the folding formed. Within the RNA, folding formed.
Yes, sir.
As a result, now this RNA polymerase cannot move forward. Stop the
Okay.
transcription. Now,
So that is inhibiting itself from moving forward, right, sir?
that's it.
All right,
The
sir.
inhibiting movement of RNA polymerase,
Yes.
right?
Correct, sir.
Right. So now transcription clear.
So why does this happen actually, sir? What might be the reason for this to happen rather than going in for the normal transcription?
Only one reason, the rich formation of uracil formation and rich formation of guanine cytosine. Here the simple
Mhm.
adenine uracil, which bond? How many bonds between adenine and uracil?
Double, sir.
Double bond.
Yes, sir.
Just write down always double bond, double bond, double bond.
Yes, sir.
Immediately it follows triple bond.
Yes. So will this be the series consecutive, sir?
Right, just wait. Here, this is the weak bond. Guanine cytosine strong bond,
Strong.
right?
Yes, sir.
Guanine cytosine strong bond. But weak bond immediately follows the guanine cytosine, guanose cytosine. Here always the guanine and cytosine try to each other. Actually, this guanine cytosine region also, for example, this is the guanine. It is cytosine. This is completely C rich, completely G rich. Between here, just uracil.
Okay.
Just imagine.
Okay.
Now,
sir.
where is the affection between guanine, uracil, guanine, cytosine? Always guanine bond with cytosine, right?
Yes, sir.
Yes,
So
sir.
that pulls it.
Right. That's why
Okay.
this guanine always try to move toward the cytosine, as cytosine also try to move towards the guanine. Due to this
Yes, sir.
mechanism, always this uracil goes up and this
Okay.
goes down. Guanine cytosine goes down and formed as a goes down and form as a loop-like
Hmm.
formation.
Okay.
Is it clear?
Yes, but the uracil pushes up, sir.
That's it.
You mean to say?
Because the uracil is weak bond,
Okay.
right?
Yes, sir. Okay, okay,
Simple,
okay, okay.
simple. Just wait.
So that uracil is from DNA? No, sir.
Just wait.
Uracil
Red
should
color,
be from our.
right? Just wait. Red color, and here green color, red color, rich guanine, rich cytosine. Between uracil. These are the
Yeah.
weak.
Yes, sir. If that is there, then the loop should go down, no, sir. The uracil should go down, no, sir.
Maybe down or up, any direction.
Okay.
But
The loop.
whatever it may be, the bond formation
Huh.
between guanine and cytosine only.
Okay. So that is
It maybe
guanine and
goes up.
cytosine will bond. Huh.
Yes. This both attached and bond formation. When due to this
Loop,
bond
whatever
formation,
in between is the loop.
that's it.
Just like the one that is at the bottom, GGAU, that one, the loop here, bulb-like structure,
Yes.
hairpin. Okay, sir.
This A, G,
Yeah.
these are just common nitrogen bases.
Yes.
Adenine, uracil, adenine, cytosine, adenine, uracil, adenine, guanine. But they are not
That
a
is
rich.
just like a normal RNA strand kind.
Right. There is no bond formation. Is there any
Yes.
bond formation
No.
between adenine,
No.
guanine?
No.
No.
So that's why this is only simple bulb-like structure. But is
Yeah.
there? It should be compulsory formed here. The triple bond between guanine and
bonds.
cytosine.
Okay.
That's why this
Got
this loop formation. This is initiate to form the loop.
Okay.
Right.
Yes.
Now,
Okay.
now
And the rest
immediately,
continues as usual.
right.
Yes, sir. Got it, sir.
So transcription process clear.
Yes, sir.
Now translation process. In translation, what happened? Please be silent. Hello, please. Please. Thank you. Translation. What is translation?
Synthesis of proteins from mRNA.
Right. We know already how the mRNA prepared. Actually, after formation RNA, there are number of changes in eukaryotes. In eukaryote transcription, after formation RNA, mRNA, that is not a completely matured RNA. After formation RNA, some processing are there. That means RNA process, 5-methyl capping, polyadenine. After this process, matured RNA formed, and also introns removed. After formation mRNA, introns are removed, and polyadenyl tail formed. Before that, methyl guanine cap formed. This some post changes, that means post transcriptional changes in eukaryote. I think not given in our book, but remember it. In eukaryote transcription, after formation RNA, the post transcriptional changes are formed.
Sir, this is only in eukaryotes, sir, not in prokaryotes, sir.
What I am explaining only prokaryote.
I mean,
What I am explained so far, that is in only for prokaryote. Same eukaryotic, same process, initiation same. But as I said earlier, but what are the enzymes involved in? Different type of enzymes are involved. and also different type of factors also involved, not only one enzyme, not only one or two factors. In eukaryote, just wait, one simple word, eukaryote transcription, the more complex when compared with prokaryote. In prokaryote, just simple process, but in eukaryote, complex process. Complex process means more cofactors are utilized, involved for initiation. But only variation in initiation. Elongation same, termination also same. But after termination, post-transcriptional changes present in eukaryotic transcription. Post-transcription means 5-methyl cap forming, removal of introns, polyadenyl tail formed. This is post changes. But remember it, no need to explain here, because in our book also, material also given only prokaryotic transcription, not given eukaryotic transcription. Okay, right? Hello?
Okay, sir.
Okay, sir.
Right. So after formation RNA by initiation, elongation, transcription on the mRNA strand, protein strand prepared. That process called as translation. And what are the requirement? The major requirement of the translation: ribosomes, transfer RNA, mRNA, amino acid. Already mRNA prepared, right? That's one clear.
Yes,
And
sir.
rest of the all present in cytosol. Amino acids in cytosol, tRNA in cytosol, ribosomes in cytosol. Let's discuss one by one. Later, we will enter into the translation process, right? First, let's see tRNA structure. A tRNA, where it is? This is tRNA structure. Be careful. If you understand about ribosome structure and tRNA structure, then only you will be able to understand about translation process. In translation also, the most important one is initiation. Like a transcription, initiation is most important, right? Before initiation, some other requirement are need. These are first, let's see about tRNA structure. Is it okay?
Yes, sir.
Right. Actually, amino acids which are present in cytosol, another requirement amino acids, right?
Yes, sir.
Okay, first of all, how the protein formed? What are the subunits of proteins?
Yes.
Subunits, protein subunits.
Amino acids.
Amino acid, right? Amino acid. That means after formation mRNA, these amino acids attach to the mRNA strand and prepare protein, right?
Yes.
Yes, sir.
But here the amino acid cannot recognize directly the sequence present in the mRNA. Amino acids never recognize directly the mRNA sequences. But with help of tRNA, the amino acids can recognize the sequence of mRNA. Therefore, the tRNA plays an important role in translation. Okay? Basic structure of tRNA appears like a clover leaf. Angiosperm plant clover. It look like a clover leaf. Look like a clover leaf, like appearance. And the tRNA is made up of only 60 to 80 amino acids. It made completely, the tRNA made up of 80, 60 to 80, most probably 80. Nucleotides made up of 80. The tRNA made up of only This one, what we given our book? Transcription. Okay, 75 to
5 to 100.
100. Okay,
Yes.
75 to 100. Just follow our book. 75 to 100 nucleotides. Right. And the first, this tRNA model first discovered by Hole et al. In 1964. The same structure was observed in yeast cell also. And right, let's see the structure. So this is the brief introduction about tRNA. Mainly tRNA, we can see clear 1, 2, 3, 4 subunits here. 4 parts. The first part, this is dihydroxyuridine. Dihydroxyuridine state. DHU loop. This one called as D loop, means DHA, DHU. DHU loop. DHU loop or amino acyl synthetase loop. Amino acyl synthetase loop. And here just 8 to 12 nucleotides are there. In this loop, 8 to 12 nucleotides there. DHU loop are called as amino acyl synthetase. Amino acyl synthetase group. Second one, ribosome attached loop. This one, ribosome T loop. Ribosome attached loop are called as T loop. Ribosome attached loop are called as T loop. Next one, anticodon region or anticodon loop. Anticodon loop. So this anticodon for what? This is mRNA strand, right? This is mRNA strand,
Sir.
right?
Yes, sir.
GAG mRNA sequence. What is the sequence of tRNA? C, U, C. G, cytosine. Adenine, instead of thymine, uracil.
Uracil.
Guanine, cytosine.
Cytosine.
Right. So this is anticodon region. And last one, this region, amino acid attachment site. This one called as amino acid attached site, and this is called as CCA end. This is called C end. CC end. So all tRNA molecule have guanine, G at 5 end. This is 5 end. This is 5 end. Guanine present. Terminal unpaired CCA end. Unpaired means.
Not paired.
So here, are you follow this marker?
Yes, sir.
This is 5 end and 3 end. 3 end, there is no pair.
Yes.
No pair. Actually, what is the nature of RNA? Is it double strand or single strand?
It is single strand, sir.
Single strand. But this tRNA appear look like a
Double.
double. In the double helical segments, guanine cytosine pairs are more common than adenine uracil. Because already told you in formation of loop formation, that means in row independent factor during loop formation, during GC rich, what happened? Loop formed. There also loop formed. Double helix loop formed due to the guanine cytosine rich pair. Due to the guanine cytosine rich pair, the loop formed. That's why it is appeared as a double helix. But actually, what is true? Is it double helix or single?
No, it is single, but due to the attraction, they are forming a bond like.
Bond like and appear as a
Appear as a double helix.
But the
Yeah.
3 end but unpaired. 3 end no stand.
The free, yes.
This free
Okay.
region actually it's CC end right here because
sir.
there are no place. So CC end, this region. Or you follow
Yes,
this
sir.
marker, this
Yes,
region,
sir.
CC end. Right?
Yes, sir.
Clear?
Yes, sir.
Right. So this unknown function extra loop, this one, unknown loop. And ribosome attached to 7 base pair here. Actually forgot. This one, 7 base pair. Here 8 to 12 base pair. Here 3 base pair. And there's 3 base pair. Rest of the base pairs are completely formed in this region. Is it clear?
Yes, sir. How many base pair in anticodon loop, sir?
3.
Okay, sir. And 7 in the ribosome T loop.
Yeah,
sir?
7 in ribosome. Here in
Yeah.
8 to 12. Right? The rest,
Yes,
these
sir.
are rest of the nucleotides. This region, this region, this region, this
Huh.
region. Right?
Yes, sir.
Now next one. Ribosome structure. First RNA structure clear?
Yes, sir.
Right. Let's see ribosome structure. The ribosomes in prokaryote, eukaryote different. In prokaryote, 70S ribosome, right? Eukaryote. Hello.
Yes,
80S.
sir.
80S ribosome in eukaryote, right? The protein synthesis occurs in specialized macromolecular complex called ribosome. This region, this subunits, this one, this large subunit, this is small. Or you follow marker.
Yes, sir.
So large subunit and small subunit, this is called macromolecular complex, and which consists of protein and RNA. In these subunits, both subunits, protein present. That means actually the protein with the help of protein and RNA, protein and RNA, ribosome form. Right? Ribosome whole chart here, no need this one. All ribosome consist of 2 subunits. 1, 2. Larger subunit and smaller subunit. And the 2 subunits reversely associated and dissociated during synthesis. Remember it. They are always associate. First, this is association, and again when forward another nitrogen bases again dissociate. Always, I will explain later in during translation process. These are always associate and de-associate during protein synthesis process. And the mainly magnesium is essential for association subunit. And in prokaryote ribosome 70S, one is 30S subunit and second one larger one 50S subunit. In small subunit consists 21 types of protein. In small subunit, this region, 21 protein subunits and 16S type RNA. 16S RNA. The larger subunits made up of 34 protein subunits. Here 34 protein subunits. Here 21 protein subunits. That means small one, already told you this is made up of protein and RNA, right? Protein, RNA. In protein, 21 protein subunits in smaller subunit and 60S rRNA present in small subunits, and 34 type are present in larger subunit, and 23S and 5S RNA present. Apart from this 23S and 5S plus 5S. 23S plus 5S rRNA present in larger subunit. Whereas in eukaryotic ribosomes are 80S with small 40S. Actually this one prokaryote, this is 30S subunit. This is sorry. $50s. But in prokaryote same, sorry, in eukaryotic same, smaller subunit 40, larger subunit 60. Larger subunit 60. The smaller subunit comprises 33 protein. Smaller subunit 33 protein. Subunits 18 is RNA. 18 is RNA. And large subunit here, the large subunit (50 proteins). 50 protein and 28 5'S RNA types are present in 68. The rRNA and protein are clustered to form subunit of ribosome. Right? Already we have seen the cluster formation of proteins and RNA in different subunits. Whereas in smaller subunit and larger subunit, the smaller subunit 21 plus 60S in prokaryote, 33 80S in eukaryote. In larger subunit 34 protein, 23S 5S RNA. In larger subunit 50 protein percentage and 28 and 5.8S RNA. So this is the components of ribosome. Is it clear? Hello?
Yes, sir.
Right. The RNA and protein are clustered to form subunits of ribosome which participated in conformation changes to move the ribosome with growing polypeptide chain along with mRNA. So for this protein and RNA mainly involved in elongation of polypeptide chain. I will explain later how the elongation takes place in translation process. Thus, these ribosomes considered as a mechanochemical systems like microtubules of flagella. That this movement of ribosome look like a flagella movement. And it's an important thing is the peptide bond formation between amino acids and ribosome is helped by rRNA rather than protein. Of course, for elongation of ribosome on the mRNA strand involved by rRNA and protein. But bond formation means here, for example, are you follow marker? Hello?
Yes, sir.
Right. So this round shape one amino acid. Like this here, number of amino acids are there, right? Like a globes. 1, 2, 3, 4, like this.
Like
okay.
chain.
Like a chain. So between the chain bond formed, peptide bond formed. First amino acid, second amino acid, third, fourth, fifth, sixth. Like for every bond formation, this bond formation activity done by rRNA only. Remember, only rRNA involved in bond formation between one amino acid to another amino acid. But movement of ribosome subunit on the mRNA strand involved by protein portion and rRNA portion. Clear? The next very important thing is the larger subunit of ribosome has a 3 sites. Ribosome 3 sites. How many sites here? Are you follow? Hello? Hello?
Only one side, sir. There are 3 sides on the top.
3 sites. The
Yes.
3 sites are first one exit site, E site, exit site. The second site, P site, peptide bond formation. Already told you peptide bond.
Mhm.
That is P site. And acyl site, A site. These 3 sites present in only subunit, larger subunit. Okay? The
Yes.
larger subunits of ribosome as a P site. P site only for peptidyl tRNA site or donor site. Donor site. That means in this site, peptide bond form between one amino acid to the another amino acid. And second one, A site. Second site is A site. And third one, exit site. Exit site means here the tRNA released from the exit site. tRNA released from the exit site. Right? This is the first site. Second, third. So remember here this is tRNA, right? tRNA exit from the E site. Clear?
Yes, sir.
Any doubt in this?
So,
I am explaining only ribosome subunits, not protein translation process. I will explain later translation. Okay?
yes, sir.
Before going to understand about translation process, you should aware about this ribosome subunit structure. What is P site, what is A site, and what is E site? Right? Clear? Hello?
Yes.
Ribosome structure clear? Any doubt in this? Hello?
Yes, sir.
I'm
Now
trying to
tRNA
understand
structure
that.
clear? Right. tRNA structure clear and ribosome structure clear. These are the major. And another important thing, mRNA, already mRNA structure, that means mRNA preparation completed through transcription process. Right?
Yes, sir.
So what are then other requirement here? What are the requirement translation? mRNA, tRNA, ribosome, both are completed. mRNA formation completed and amino acids. These
Yes,
amino
sir.
acids formed in cytosol.
Cytosol.
Let me complete in general brief explanation about translation process. In translation process also, like transcription, 3 stages. What are the 3 stages? What are the 3 stages?
The closed complex.
No, I am asking
sir.
about the transcription 3 stages. First
Oh.
transcription 3 stages.
Initiation.
Initiation, elongation, termination.
termination.
Same. Here also initiation, elongation, termination. In initiation, to initiation, what are the requirement for the initiation of the translation required? tRNA, mRNA, amino acid. Now tRNA completed, right? mRNA completed,
Yes, sir.
right? Ribosome also required, ribosome structure also completed, right?
Yes, sir.
Now the amino acids. Where the amino acids found?
It's in the cytosol.
In the cytosol, but in cytosol they are inactivate state.
Mm-hmm.
Before attached to tRNA, they should be activate with help of ATP energy. ATPase enzyme. Due to ATPase enzyme activated amino acid. Then these activated amino acids attached to the tRNA. Now this tRNA carry the amino acids towards the which region? Where amino acid goes?
To the ribosomal site, sir.
To the ribosome site or to the mRNA site. Now this attached tRNA of attached amino acid tRNA goes towards the mRNA, right? Later
Yes,
this ribosome subunits attached. First smaller subunit attached, later larger subunit attached.
Mhm.
Next, remember it, first amino acid bind at P site. This is P site, right? Hello?
Yes, sir.
First bind at P site.
Mm.
So this is second sequence. Second amino acid, this is the first one. P site. Rest of the all amino acids tRNA bind with only A site. Bind with only A site. This is very, very important. So first amino acids only, first
P.
of all, this is the first step.
Hmm.
A, E empty, SL empty, P filled up.
Yes.
Next, the coming tRNA amino acid complex attached at
P
A site, SL site only.
Hmm.
Later shifted to P site.
P site.
Meantime, the P site tRNA shifted to exit site.
Mhm.
From exit site, when this tRNA, that means first tRNA complex, tRNA shifted to E, this place vacate, right?
Yes, sir.
Now this fulfilled by second tRNA shifted to P site. What about A site?
The next one will take.
Right, the next third amino acid, third tRNA complex amino acid bind at A site. From the exit side, first tRNA moved
Moves
out.
out.
That's it.
But sir, why is that it's first attaching to P site rather than A site?
I will explain in translation mechanism.
Okay, sir.
This is just only brief introduction about translation.
Okay.
Sir, always the first amino acid should be methionine, right, sir?
Yeah, methionine. Because AUG first codon, right?
Yes, sir.
AUG,
So here also methionine comes and attached to the P site, sir.
UAC. So this UAC only code for methionine, not for other amino acid. So where the tRNA UAC anticodon present, this UAC uracil adenine cytosine only code for methionine.
AUG.
Right, only code for methionine, but not other amino acid. That's the first only methionine attached to first site. Right. Next, where some other GCC, whatever it may be, it code for other alanine, prolanine. So different type of amino acids also there, and different type of tRNA also there. So different type of amino acids attached with different type of amino acids, and this brought towards the mRNA or ribosome complex and form the bond formation in P site. This is the first amino acid, second, third, fourth, like this number of chain is formed.
Sir,
After
I have a
chain
doubt,
form,
sir. In tRNA structure, the upside it is having the amino acid binding site, right, sir? At the
Yes.
3' end.
Hmm.
So that amino acid is activated or inactivated amino acid?
Activated.
Activated amino acid.
Yes. I will explain clearly the formation of tRNA amino acid complex and activation of amino acid. There are different type of steps in initiation.
Okay,
Amino
sir.
acid activated, tRNA complex formation. In tRNA complex formation, the first 43S complex, 80S complex. So different type of complex also there. But in my next class, in detail, I will explain in detail in my next class.
Okay, sir. Okay, sir. Thank you.
But so far clear,
Yes, sir.
a brief introduction about translation also clear.
Yes, sir.
So after this is elongation region. When reach the nonsense codon, UAG, UAA, UGE, right?
Yes, sir. Amber, opal, ochre.
Right. When it is reached this region, when the P site reached to the any nonsense codon, this nonsense codon not code for any amino acid. That's why not code for amino acid. That's why the translation process will be stopped. Right? Clear? Hello?
Yes, sir. Yes, sir. Yes, sir.
Already this prepared amino chain released out.
Yes, sir.
So this is just a brief introduction about translation. In our next class, we will discuss clearly about initiation process and what are the activation amino acids, tRNA complex form, tRNA amino acid complex formation, 43S, 48S, 60S, 8S, how they are found, what are the role in this mechanism. Okay? Right?
Okay, sir.
Right. Any doubt? Today class, any doubts?
Not as of yet, sir. Once again, if I start reading, then
Please,
definitely it will be.
Please prepare notes.
I'm doing that, sir, simultaneously. But as I'm like reading, doubts keep erupting in
If
my
you have
head.
any difficulty, again, I will explain in our practical training classes. Some complex classes like protein synthesis and whatever may be protein synthesis, and also another complex classes that is photosynthesis, Krebs cycle, ETS cycle. If any complex matter, I will explain clearly. That means I will give the notes, the
That would
detailed
be
notes.
great help, sir.
Sure. But meantime, you have to prepare notes clearly.
Yes, sir.
Okay, right.
Thank you so much, sir.
Right. Can I leave?
Yes, sir. Thank you so much. Good night, sir.
Right. Bye.
Good night, sir.
Thank you, sir.